Imagine if you could look at a random splash of seawater filled with tiny extraterrestrial creatures. We could find something as wonderful as this in these plumes. A chance of a super positive outcome, so how does that influence how we can explore? Cassini has sampled the plumes already, flying through them, with no chance of contaminating the oceans, a gold star for planetary protection. So this is something we know we can do. Enceladus is a sure thing, as its plumes erupt continuously, feeding Saturn's E-ring. Europa's plumes are much more uncertain. They may be sporadic, like this Icelandic geyser, and fall back to its surface right away in the higher gravity, making it more of a challenge to sample them. The plume Hubble saw could be the result of a large meteorite, and when Enceladus passed by Europa on its way to Saturn, then it saw no sign of anything, and it would have been able to spot the likes of this very easily. So I'll focus on Enceladus for now, but this also applies to Europa if we can sample its plumes. The ocean is kept liquid by eccentricity tides. Enceladus can't go into a circular orbit because of resonances with Dione. Its heat budget is a long-standing puzzle. Some models work, but we don't know if they are valid. Evidence from earlier frozen-over tiger stripes suggests it may be freezing slowly. We don't need to worry about the next few thousand years, but the geysers may stop within 200 million years. The same geological evidence suggests its ocean is at least 1 billion years old. Its interior is probably like a carbonaceous chondrite meteorite, unprocessed early solar system material, rich in organics. That's great for life, except that it's also completely undifferentiated and porous. It's thought that all the salt from its interior has already flowed into its ocean, so it may have reached chemical equilibrium, and any life there might have run out of food and be extinct or sparse. On the other hand, tidal effects might easily supply enough energy for a complex ecosystem. Cassini's measurements suggest two main possibilities: a very alkaline soda lake or less alkaline white smoker type hydrothermal vent with hot rocks. Or its oceans might be young, only 1 billion years old, or perhaps life evolved more slowly. If so, perhaps it is only the early stages of life or no life. There's a tendency to look at this and say the right-hand side is what astrobiology is all about, complex life. And in the case of the left-hand side, let's leave it to the planetary geologists. And for that matter, we might even feel that we have a duty to bring Earth life to a lifeless ocean. I'm going to suggest that Earth life or non-life is also of extraordinary interest for astrobiology, and as well as that we have nowhere near the understanding needed as accidental or intentional ocean eco-engineers. This visualization shows how Earth life makes protein. Here, messenger RNA produced by DNA polymerase meets a ribosome, a huge molecular machine that spontaneously assembles around it. These green triangular transfer molecules each bring a single amino acid to the emerging protein chain. You can see the amino acids as red dots at the tips. This is going on at this speed in every cell of every known Earth creature, and the visualization just hints at the complexity of the process. It works, but it seems Heath Robinson, or Rube Goldberg if you're from the States, like the professor's potato peeling machine. Life must have started simpler. Perhaps as a self-replicating polymer not yet alive but encoding information, or perhaps as an autopoietic cell. This means it takes in ingredients, uses them to recreate its metabolism and cell wall, and expels waste. It's only a dozen or so chemicals, and it just splits when it gets large enough, or a daughter cell from inside escapes. This shows time of origin against gene complexity on a log plot. Prokaryotes, cells without a nucleus, are oldest. Continuing the line back in a straight line, and it crosses the axis at around double the age of the solar system. So, perhaps life started on an exoplanet around an older star. This passes through a nebula where our solar system was forming in one of these fingers at the tip. It misses most of the embryo stars, but by chance comes close enough to ours to transfer life by meteorite impacts. You can do this from quite a distance. Here, Earth could be forming in one of these dark bands. Or, the solid line may show the slope for DNA with error correction. Perhaps earlier life evolved much more quickly, as in the dotted line. Life in these extraterrestrial oceans could be anywhere on this graph. The oxygen-rich ocean of Europa could even have the likes of fish and octopuses. It's unlikely we have both reached exactly the same stage, Europa life and Earth life. Modern multicellular life is quite recent, so it might not have evolved in here yet. Or it might have evolved to some future form of life that we don't have yet. Perhaps 50-50 either way. And if it got that far, what about a civilization, millions of years old? In an ocean with no fire, their technology would be limited. They would know nothing about us, and we would know nothing about them yet. In the other direction, it could be somewhere in the first half of this graph. More than half of evolution is hidden from us according to this graph of complexity. Each pixel in this graph covers over 70 million years, so our lab experiments like Yuri Miller explore a tiny fraction of the first pixel. That leaves a huge gap, which must have steps as momentous as the development of a nucleus, multicellular life, and the plants and animals. But what those are down here, we can only guess. In some ways, that might be the most interesting outcome of all, to fill in a little of this gap. That's why I suggested an early or non-life ocean could be just as interesting for astrobiology. There's also the possibility that it's a different kind of life, and that's true for the entire graph. If life evolved around a previous star, there's a chance it's related with a common ancestor older than the prokaryotes. But if the ocean is young, so that any previous life deteriorated, or if the life evolved quickly, it's likely that there's to be independently evolved, especially as there's little meteorite transfer between Enceladus and Earth. Earth life seems optimal. It's stopped at 20 main amino acids after all, and that's with a lot of spare coding capacity. Here we can see that our serine, for instance, is coded in 4 different ways. Each of these could have coded for a different amino acid. However, this could easily be a local optimum. Let's think of the height as anything you want to optimize, such as the efficiency of the metabolism of the entire cell. You start in Australia and keep walking up, and eventually probably just get to the top of a small hillock. If you allow yourself to go up and down and travel great distances, the highest peak you can reach is only a little over 2,000 meters high, less higher than half the height of Mont Blanc. If you were at the very top of this peak, because it's a summit in this analogy, any tweaks, such as adding more amino acids, will always take you downhill. So you might well think that Earth life is the best that any life could possibly be. But if you started in North America, you'd end up at the top of Mount McKinley, which again would be a summit that seemed totally optimal. Everything you try takes you downhill, unless you were able to explore as far as South America. And then, both of those would be a lot lower than Mount Everest, which you reach as you gradually explore Eurasia. So there may be many different optimal best solutions for life, and we only know of one. Here we are looking at the workings of the oldest cells, such as the prokaryotes. So, what's more, life on Enceladus could be better than us at metabolizing. It could be the Mount Everest of metabolisms. Or Earth life could be Mount Everest, and Enceladus life is relatively feeble. Since we're just looking at the interior of the early cells, there's no way of knowing. If Enceladus does have life at all, it's perhaps 50-50. Specialists who study the interior of a cell sometimes compare the complexity of the interactions to an ecosystem. In this analogy, all Earth life has the same ecosystem inside. I've listed a few of the things in it. Experts would add many more, common to all Earth, interacting together in complex ways. This ecosystem is so similar, life forms often swap genes in parallel transfer, and they still work. Here's a gene from a fungus. Gave this pea aphid the ability to generate carotenoids, which turns it red like a carrot. Imagine if the African savanna, in the same way, was the only ecosystem biologists knew about, that all ecosystems were minor variations on it, and then one day you discover a coral reef. We have never seen a tropical rainforest or tundra or the seashore or anything. This shows how much we could learn just from observation of a single cell or protocell based on different principles. So of course we are careful This is one of the Viking landers' gold standard of planetary protection on Mars. Nevertheless, you can question if it was sterilised enough. The aim was to have no more than a 1 in 1,000 chance of contaminating Mars during the exploration stage, which is how they were thinking about it then. Is that enough, given that it could be a super positive outcome? Anyway, that decision is made, and it is generally thought that we have not contaminated Mars with Earth life, mainly because conditions there are so harsh. But it's important to know that we haven't yet looked at our spacecraft on Mars to see how many dormant microbes there are, and in what species. Our calculations predict that there are microbes, and that they are sitting there doing nothing. Again, we don't know for sure, because we haven't checked to see if our spacecrafts are replicating Earth life around them. For instance, Phoenix was crushed with the advancing winter dry ice sheet. We have no experimental data from Mars on this, on what happened to the microbes on it. So this is a little bit shaky. We don't think we have contaminated Mars, but it's not the best basis for making decisions about icy moons. The National Research Council looked at it, and especially since these oceans are much more habitable in Mars and probability assessments are more uncertain here, this approach just wouldn't work. They recommended binary decision trees. The orbiter or flyby passes at stage 5 with a yes, because it's not going to encounter a habitable region, but any Enceladus life mole, or penetrator, or cryobot passes through all the way to the final stage. And then you have stringent planetary protection or cancel mission. The Russians in Antarctica drilled nearly all the way down to Lake Vostok and then discovered they were approaching a lake. So they just stopped for over a decade. Eventually they drilled into it in such a way as to trigger a mini geyser of 30 or 40 meters so that there would be no forward contamination into the lake. This is like a flyby mission. So far they have only sampled this plume. It would be easy to send a submarine down, but they feel they haven't yet got the technology to sterilise it. So in the same way, when exploring Enceladus, we should always have the option cancel the mission, not ready yet. This rabbit-proof fence was built to protect Western Australia in the 1900s from rabbits introduced by European colonists. It was the longest fence in the world, and it did work after fashion for some years. Cartoonists had a field day, showing here a probable use that rabbits might make of it. I wonder what kind of fence we could put across the Enceladus Ocean. Busbar recommended a target of a chance of 1 in 10,000 of contamination in the next 1,000 years. However, there was a lot of discussion in the workshops. There is no science you can do, no a priori calculation, no experiments that can give you these two numbers. It is an ethical decision. If you think about it as a potential super positive outcome, you might look at these numbers a bit differently. We have a chance of a super positive benefit to billions of people, revolutionising perhaps medicine, agriculture, nanotechnology. We don't know how probable it is, but it is a possibility. In the direction of a sample return to Earth, the legal situation is clear. This is a principle of modern international law introduced because of the new things we can do. A sample return could, in the worst case, cause degradation of the environment. For instance, if we return a life form better than Earth life, then that could degrade the environment. In a situation like this, we must take precautions even if we don't know what is in it. It's no good saying, I have no idea but surely it's okay, or use hand-waving arguments. We have to take precautions anyway. And it's us, the proponents, that have to do these science, and the discussion has to involve all the affected parties, which in this case may mean the entire world. We can never say we are the scientists, we will make the decision for you. That's totally wrong. And you have to consider all possibilities, including always no action. That can get forgotten. If you ask experts, how can I return this sample safely? That's the wrong question. They must always have the option to say it is not safe, yet, if that is what their research shows. I suggest that it should be the same for super positive outcomes in the forward direction. You should take precautions even though we have no idea what is there. As proponents, it's up to us to prove that we are doing is not going to impact on it. And I would say, because it is super positive, it impacts on the entire Earth, because of those billions that would be affected in a positive way in the best case. So it should be similarly open and involve discussing all possibilities, including again no action. Here I have just adapted the first sentence of the wingspread text, one of the statements of the precautionary principle, to show the idea. So I've changed harm to health or environment to super positive outcome, and you get a reasonable guideline, I think, in the positive direction. Carl Sagan once said about a sample return from Mars, you can't take a small risk with a tiny life, with a small risk with a billion lives. Nick Bostrom has developed a philosophical approach to existential risks. That is to say, risks of serious impact on human habitability of the Earth and our future life prospects. He has suggested that we haven't evolved an intuition for these risks because we haven't encountered them before. He suggests one way to get a clear idea is to look at the expected number of people affected. So, for instance, in sample return discussions, they often say that the risk of 1 in a million is acceptable, a tiny figure used for biosafety laboratory designs. But if you multiply that by the population of the Earth, the expected number affected is 7,300 people. That becomes millions or trillions of people if it is possible that all future humans could be affected. So even with a tiny chance like that, sometimes the expected number impacted can be huge. I suggest the same applies in the forward direction. There is absolutely no impact on human health, but if you increase the chance of a super positive outcome by 1 in 10,000 by taking more care, and do the same calculations, the expected number is 730,000 people benefited. And again, it becomes billions or trillions of people benefited if you consider future generations. Here, possible future beings from Earth study Enceladus. At bottom left, H.G. Wells' human for 1 million years in the future, little more than eyes and a brain, because machines do everything. Then, the humanoids from Avatar, standing for the idea of future humans more closely connected ecologically with the Earth. Then, there's Alex the Grey Pigeon, the Grey Parrot, possibly the most intelligent non-human known. Perhaps in the future, we may get civilizations of descendants of parrots and other creatures either coexisting or following on. Over the next 500 million years, surely thousands of such civilizations will arise and fall, and from time to time some of them will visit Enceladus. We have a super positive outcome for all of them, not just ourselves. Also, of course, for any beings in the oceans already. This is the next step up from a flyby, an orbiter. In the plans, it starts with several gravity-assist flybys of Titan off the picture, then the inner moons, ending up in an equatorial orbit around Enceladus with almost no delta V. Sadly, a polar orbit around Enceladus is unstable, so it can only pass through the geysers at its south pole occasionally. But the big advantage is that it can capture the material at around 100 meters per second, and is only 8 times faster than Usain Bolt. With capture into aerogel, we can hope to find even viable microbial spores and intact autopoietic cells if they exist. You might think that every sample would be the same. However, we'd be able to fly at different heights. For instance, we can go lower for the larger particles, which Cassini has already done. Also, we can go through the plumes at different states of the tides, which vary all the way around Enceladus' orbit, and we can sample them also individually. And if there is life here, perhaps sporadic subsurface events, equivalent of algae blooms, might send interesting material into space from time to time. You get a rather poor idea of what we can do in situ from our mission so far. Only one of these instruments has ever flown, the Philae Cossack chiral cask chromatography. We can now send sensitive labs on a chip, able to distinguish just one molecule of an organic in the sample for the subcritical liquid water extraction. This is now miniaturized right down to the astrobionibbler. Monoclonal antibodies can also be very sensitive. Solid 3 in this instrument in its test in the Arizona desert found a previously undiscovered habitat 2 meters below the surface. It's so sensitive. Incidentally, it is not limited to Earth-based organics. The key to Cassini's success was to send a wide range of in-situ instruments. Nobody expected the plumes, but it was so well equipped it had what was needed to get started. I suggest we do the same for our search for life. This can help invigorate the entire field. Many teams of astrobiologists will see instruments fly in space for the first time since Viking. And even if they don't find anything, for instance, if the miniaturized informational polymer sequencers don't find DNA or XNA, this may tell us that it is not related to Earth life and rule out other alternatives. And if it does find them, it sequences them. Or, if the microbial fuel cell finds no redox reactions, another result can be very significant. Many of these instruments are so small, far smaller than for Viking. Indeed, many of them are labs on a chip now. We could maybe even send all of these. I would suggest a good high-res optical microscope as well. That's just because how silly it would be if there was life in our sample moving around purposefully and we just couldn't see it. This shows the next stage. You might think that there is no chance of contaminating the ocean with an ice mole, because after all the geysers erupt upwards. But unfortunately, the cracks continually open and close with the tides on every orbit. Also, new geysers must form from time to time. It could get crushed or work its way down, or parts of it could. Notice also that the interior of the geyser is a near vacuum filled with water vapour for a long way down. How we sterilise it may depend on what we find. For instance, if the ocean consists of autopoietic cells, we may need to take great care not to introduce even dead machinery of Earth life, in case the cells take it up and learn new tricks. Or, if it is closely related to us, we may need to take great care about GTAs, which can transfer genes rapidly, even overnight in seawater, to completely unrelated phyla of archaea. With ideas of a super positive outcome, we need to sterilise the ice mole totally, not just for the next 1,000 years. I think that could be feasible. With ExoMars, we're exploring carbon dioxide snow after preliminary cleansing. At temperatures above 31.1 degrees centigrade and high pressures, CO2 becomes supercritical, a state between a gas and a liquid, penetrates cell walls and dissolves organics readily. When pressure is reduced, the organics are blown away in the snow by hot dry air, leaving nothing behind. We would need to sterilise any electronics at the end of the mission, perhaps by the same method as it has successfully sterilised USB drives. Or perhaps we could do it with ionising radiation. Even if we solve the sterilisation issues, I suggest our first mission looks at the plumes with as wide a range of instruments as we can possibly send there. Because until then, we can't do an optimal design for our ice mole, and we won't even know what are the best instruments to put on it. If we get interesting results, it will surely be easy to get funding for follow-up missions. By then, our spacecraft may be able to get there quickly, drill more quickly, return samples sooner, and have smaller and better instruments. As for sample return, that needs a new talk. But in short, you might be surprised at how complex the suggestions are for planetary protection for a return to the Earth's surface. It would add over $500 million to the cost, and may still leave some safety questions unanswered, as well as requiring about a 10 years process of new internal and international legislature. I think the safest, simplest, and also lowest cost place to return it to would be to a tele-operated facility in high Earth orbit, which humans go nowhere near. I have detailed reasons for that, which I can explain separately. Anyway, we wouldn't know for sure how to design the return capsule until we know what we need to return, where to return it from, and how to best preserve it on the long journey back. So instead, I suggest we get the ingenious teams of astrobiologists at this stage to submit their ideas for instruments research in situ, and send the very best of these on our next mission to look at the plumes close up. Meanwhile, of course, we continue with our research into the ice moles and the penetrators and so forth, because we hope we'll be able to send those in the fairly imminent future after we've found out the results from the in situ researches.